why I told you about the importance of that syllabus. In the syllabus, there is a topic that is called chemistry of proteins, chemistry and metabolism of proteins and immunology. And BC 5.1, this is basically deals with the chemistry of proteins. You can see that, and you can read, discuss briefly the structure of amino acids and classify amino acids on the basis of nutritional and metabolic significance. It will take half an hour to cover what you will read at the end, but for this I have to take 5 classes. Why? Because questions will not be asked only on this, and that's what I have seen also. This is much more beyond the syllabus, and to understand this thing, you need some more information, additional information, and those additional information, if these are not given at this stage, you will not be able to follow the subsequent classes. Okay. So, first there is a issue that briefly structure. So, I will discuss briefly the structure, because structure you cannot remember, but this time again I have seen the examiners have written the structure and asked the students what is this structure. So, which examiner will come, what will be his intention, I do not know. So, better to be aware of. At least I know one of my junior who came from PGI Chandigarh is very fond of asking the structures. Classify amino acids on the basis of nutritional and metabolic significance, I will definitely tell, but in addition I will tell something more. So, my point will be, what is an amino acid? I cannot use the board, that's because of this thing. So, what is an amino acid and how many amino acids are there? What are the 3-letter and single letter abbreviation of amino acids? Classification of amino acids, what you said, what you have read, that your syllabus is on nutritional requirements and metabolic significance. But there are other classifications also that I will discuss with reference to structure. And then these points, if I do not tell you that enumerate the major physico-chemical properties of amino acids, you will not be able to understand the protein also. So, some part I will tell you, and some of the things which you will be also using in your practical classes, and this will come as an MCQ also. And of course, biochemistry subject is taught in general colleges as biochemistry, in botany, in zoology, in veterinary, wherever life is taught, biochemistry is taught. But you as a medical person must know its relevance within the human body and must know its biomedical, because you are, after all, you are a medical student and you will be dealing with human beings, not with animals. Or clinical relevance. Biomedical and clinical is not synonymous. Clinical means what the patient will come, what you will see in the clinics. And importance, of course, these are the topic, everything is important. Every organ in our body is important. Eye is as important as brain or kidney or heart. Even the anus, if you say that through which we are passing stool or through urethra through which we are excreting, if that is closed, can you imagine what will happen? You are going to die simply. So, no organ is bigger, no organ is greater. So, every organ has its own importance, including its position. Suppose if my eyes are placed here, do you think it will be proper? No. Do you think if I see one eye with one eye, it will be proper? No. So, what 100% functioning of the body is required. So, every importance is there, that I will discuss. So, what is an amino acid? Amino acid, remember. So, amino group will be there. So, that NH2 group will be there, and acid means COH group. So, any organic compound which will have an amino group and a carboxyl group is basically an amino acid. Okay. So, What is the difference between organic chemistry and inorganic chemistry? Can anybody tell what is the difference between organic chemistry and inorganic chemistry? Follow. Jore bolla, others will be able to hear.
So organic chemistry majorly deals with compounds made of carbon, hydrogen, oxygen,
Carbon
hydrogen, and their other derivatives.
or organic chemistry?
So organic.
Organic chemistry,
And inorganic deals with other elements.
other elements, not carbon.
Majorly, organic chemistry deals with compounds which are majority is carbon and hydrogen.
Organic chemistry includes carbon also. Other compounds will also be there. But inorganic will compound almost without carbon. That is the difference. And what is the difference between the organic chemistry and a biochemistry? Sit down. You must understand the difference. What is the difference between organic chemistry and biochemistry? Because organic chemistry is told in the chemistry branch, main branch, but you are here with biochemistry. Organic chemistry and biochemistry will deal with the same organic biomolecule. One thing will be the living. Biochemistry deals with the chemical events occurring in a biological system. Once the person is dead, organic chemistry will be there in the dead person. Okay, it is not that organic chemistry will not be there, but biochemistry basically deals with the living, any organism, not human being. So, in carbohydrate, you have been told, carbohydrate, have you attended a carbohydrate class? Okay, carbohydrate means carbon, hydrogen and oxygen. So, in addition, nitrogen is present in protein or amino acid. What are amino acids? Amino acids are basically building blocks of protein. Means if you have a protein, if you break it down at one stage, you will get the smallest, just like a cell, unit of our cell of our body, you will get an amino acid. If you break down carbohydrate, at the end you will get a monosaccharide. So, amino acid is the building blocks of proteins, and it also performs several metabolic functions that also I will discuss during each amino acids, I will tell you. So, by and large, the amount of nitrogen in protein or amino acid is fixed, that is 16%. So, that 16% you must remember, and as I told you, these amino acids are the building blocks of protein. Basically, proteins are polymer of amino acids. Carbohydrates are polymers of monosaccharides. And all the amino acids in the human are in the form of L-alpha amino acid, not in the humans, I would say, all the proteins, because it has been designed so. What is alpha amino acid? Alpha carbon is there, the carbon to which both the amino group and carboxyl group are attached, it is called the alpha carbon. That is the reason its name is alpha amino acid. Okay, amino at alpha position. And why L? Because of the optical activity. Next slide I will show you why it is called L. So, you know by this time that there are 20 amino acids, but in the nature, which converts basically the entire nature, there are 500 amino acids, but we do not have codons for 500 amino acids. We are having only for 20 amino acids. Okay. So, what are the roles of those amino acids and what do they do? Because they are produced in the nature as a byproduct of the metabolism, or at times the several organisms produce these chemicals as a defense mechanism to protect themselves or as an excretory product. So, almost more than 500 amino acids are available in the nature, but only proteins are synthesized exclusively from 20 amino acids, and that is alpha amino acids, L-alpha amino acid, remember L-alpha, and these are encoded by codons So, what is an alpha carbon? Alpha carbon is the carbon to which amino group, carboxyl group, hydrogen atom and a unique side chain. This is a unique side chain. If a board was there, I would have clarified a little bit better. There is R group here. This is unique feature of the alpha carbon. This R can be H. It can be hydrogen, that's the minimum, or it can be a R group as such. And this R group basically determines the entire characteristics of the different types of protein, amino acids. Now, asymmetric carbon, what is an asymmetric carbon? Asymmetric carbon means around one carbon, there are 4 different types of groups are attached. Then it is called an asymmetric carbon. Suppose here you see, look at this structure, the 4 groups are there. So, carboxyl group is there, amino group there, hydrogen there, and R is there. By chance, if this R is H, that means there are 2 H. So, there is no asymmetric carbon. Okay. And then there is a L form, as I told you, L amino acids, and D form. So, this is optical activity. There is a polar emitter machine through which a polarized light is passed through. So, because of the property of the molecule, the right will be either rotated to the right or to the left. When it is rotated to the right, it is called dextrorotatory D form. When it is to the left, it is called levorotatory. So, that polar emitter machine is not there, otherwise I would have demonstrated to you. It is a complex, physics is involved into it. So, only thing is remember that the L amino acids are present when this into remember very easily, when the amino group is on the right side, it is called D form, and where it is amino acid in the left form, this is L form. And in all your subsequent protein or amino acid classes, you will see that COH group which is written here will be written in this side. So, that will be the C terminal of the amino acid or carboxyl terminal of the amino acid, and this will be the amino terminal of the amino acid. So, these all the amino acids are in the natural, which are present in the protein, as I told you, it is in the L form. It is in the L form, and D form is an unnatural form, but there is no watertight. I am telling you, there is nothing called 100%. Our knowledge is expanding, and whatever you are reading at this stage, at least what I read in 1981, there is a sea change what I read in 2025. So, many new things are come, just like the D form. The free D serine and D aspartate are available in the human brain tissue, which are not known earlier. Similarly, D alanine, D glutamate, they are present in the gram-positive bacteria, and D leucine, D methionine in Vibrio cholerae, they are causative organism for cholera, and there are several antibiotics in which the amino acid is present in the form of D form of amino acids. So, L form is only for formation of protein. Okay. So, classification is on the basis of essential amino acids. Okay. So, what are essential amino acids? There are 20 amino acids, remember. First, 21 and 21st and 21st and 22nd is there. But when a question is asked, usually the 21st and 22nd is told, not forgetting all those 20 amino acids. But remember, by and large, 20 amino acids are there, and codons are there for 20 amino acids. Okay. So, when I was class 2 student, I used to remember that essential amino acids in this way, that is Van Fleet. There are many mnemonics which are available in many books. Whatever way you think, you make it, because there are many things you have to remember. You can remember one line of one song, one quotation, and then you can memorize. So, Van Fleet was my abbreviate mnemonics because the mnemonic term was not known to me at that point of time. So, you can write this thing, there are other things, other mnemonics also you can remember, but this all 10 amino acids, the V is for valine, A is for arginine, M is for methionine, P is for phenylalanine, L is for leucine, another L is for lysine, I is for isoleucine, T is for threonine, and T is for tryptophan, and H is for histidine. One thing I am telling you, there is one T here. One T is at this tyrosine. So, do not get confused with this thing, because tyrosine is why I have written from phenylalanine tyrosine can be produced. So, that is why tyrosine is not an essential amino acid. So, in our body we are having an enzyme called phenylalanine hydroxylase. That enzyme converts phenylalanine into tyrosine. So, that is the reason tyrosine is not an essential amino acid. So, that is why I do not in this TT forget about tyrosine. Only threen and tryptophan you write. So, out of these 10, 2 are called semi-essential. Basically, these are all essential. We do not have the machinery. The reason what were described about the semi-essential nature, because we cannot produce them in adequate amounts during the periods of increased requirement, like during infancy or recovery from injury, recovery from illness. Suppose a tissue has been lost and some good amount of protein is to be synthesized, that part is not possible during pregnancy when fetus is growing, placenta is growing, or during any kind of increased demand of the body cannot synthesize adequate amount, but arginine is required only during infancy. But we do not have any mechanism for histidine synthesis in our body. So, histidine is by and large, it is an essential requirement, and Harper is considered the standard textbook. I checked with Harper, although names are not there, but this list of these 10 amino acids are categorized as essential amino acids. There is no term called semi-essential in Harper books at least, which is well regarded. And rest of the 10 amino acids are non-essential. So, only thing remember, this arginine and histidine can be called as a semi-essential because they cannot be produced in adequate amounts during periods of increased demand, particularly arginine during infancy, but histidine can be produced in our body. It cannot be produced. Arginine can be produced through an intestine-kidney axis. A part of the precursor of arginine is produced in the intestine, and rest is done by the kidney. Second part was that metabolic significance, that was under classification. So, what is metabolic? There are 3 things. One is ketogenic, one is glucogenic, one is both ketogenic and glycogenic. Glycogenic or glucogenic, glycogenic I should say, glycogenic, glucogenic you can say also. Glucogenic will be the most appropriate term. Ketogenic means leucine. If you say, if you are given a choice, leucine will be the choice, but lysine is also ketogenic, means at the end of metabolism, you will get an acetyl-CoA acetoacetate, and these will form the ketone body. They can form also fatty acid. Okay. So, from leucine and lysine also categorized both as glucogenic and ketogenic. And again I have made one, I used to remember because LIPTT, that is lysine, isoleucine, phenylalanine, tyrosine and tryptophan. These are both ketogenic and glycogenic. And rest 14, that means we have covered here 5, 6. So, rest 14 are all glucogenic amino acids. From at the end, from these amino acids, glucose can be produced. But from glucose amino acids cannot be produced because these 4 has to be supplied Since body doesn't have any mechanism to synthesize those, we have to take it from the diet. And if we do not take it from the diet, protein synthesis will not occur because these enzymes will not be incorporated into that protein. So, there will be diseases. Okay. So, as I told you, essential, why it is essential? Body cannot synthesize. This is the simple purpose. So, we have to take it from the diet. Semi-essential or conditionally essential, as I told you, it is not in always in sufficient quantities to meet the increased requirements. Or non-essential means body can synthesize. As I told you, tyrosine can be produced from phenylalanine. So, there are metabolic pathways through which one amino acid can be converted into other or from all other sources. So, 3-letter and single letter abbreviation of amino acids, because all the amino acids you can see this the number of words are big, larger. So, that there is a small form how this can be represented is basically the purpose of having a 3-letter abbreviation. And you see that 3-letter abbreviation, these the first 3 word letters are used, glycine. Okay. Historically, I am telling you, among the 20 amino acids, it was asparagine was discovered first from a fungus, that is aspergillus, and then in glycine, there is cysteine was discovered in 1810. But cysteine is basically a dipeptide which comprises 2 molecules of cysteine. Okay. So, if you look at this thing, leucine, LEU, CIS, methionine, MET, but you see the isoleucine here, it is not ISO. Okay. Proline, again you see the red letter marked with tryptophan, TRP. So, asparagine is termed as ASN, isoleucine ILE, glutamine is GLN. Why glutamine? Because glutamic acid is there. You see glutamic acid, glutamate, GLU is there. So, for glutamine, you cannot use the same 3-letter word. See why TRP? Because tyrosine is there, TYR, so RY you cannot use. So, tyrosine, tryptophan is used that as TRP. So, why 3-letter is important because when we denote the amino acid sequence, if we start writing all the amino acids subsequently, it will be very long. So, just to, if you use that protein or peptide sequence, that phenylalanine, PHELA, VAL, ILU means you understand that indicates that amino acids. Even if there is single letter is used, one is you just note down this one, KDAEL sequence of protein. You note down this one. KDEL sequence of protein. What is K? K you see what is lysine. A, D, see D, look at the D, D is aspartic acid, E is glutamic acid and L is leucine. KDEL sequence is important when you see in a protein at the terminal part, that sequence is there so that the protein is targeted into the endoplasmic reticulum or Golgi apparatus. So, once the protein is synthesized, protein is targeted, sent to either within the cell to other organelles, can be in the cytoplasm or cytosol, or it can be exported. By chance, if the protein is sorted not into the Golgi apparatus and the KDEL sequence is there, it will be transferred back to endoplasmic reticulum. So that KDEL sequence, that is the importance of KDEL sequence. Why single letter then? See, again if you write 3-letter, then we will take a space. There is one lady called, it is a great person, he is a biochemist, biophysicist and a computer expert also. See, computers what we were seeing in India in late 80s, computers were available. And she died in the year 1983. bioinformatics term was coined in the year 1970. Today you know, many of you know about bioinformatics, but see what in 1966 on bioinformatics about the sequencing of protein. So, unlike today's mobile or computer, storage capacity of the devices were very little. So, there is a punch card was there. So, limited storage capacity was there, for which single letter, this part is not written in your book, you just listen. So, punch card was used, for which a limited storage capacity, small letters were required. So, that is the reason why single letter abbreviation was there. And now you note down these two parts. This del F508, you just phenylalanine, you see the single letter word letter is F. Okay. So, del 508 mutation of CF gene, that is represented deletion of phenylalanine at 508th position of this one CFTR gene. The what is CFTR? And you will be told once again that basically cystic fibrosis transmembrane conduction regulator, that is a protein. That protein is used for transporting chloride, that is a chloride channel protein. Now, if there is a defect, that phenylalanine is not there, if there is a deletion, there will be defective protein and disease will be caused. So, this mutation delta 508 mutation can be represented as this delta just like a triangle F508 or F just F508 del. F means now phenylalanine. Okay. So, KDL sequence, as I told you, it is theta amino acids for signaling of protein within the endoplasmic reticulum. If you arrange the single letter, basis of single letter, why the single letter was used. So, if we arrange in alphabetical order, you see that alanine is there, arginine is there, aspartic acid is there, asparagine is also there. Now, how will you do, use the single letter? It is difficult. Similarly, cysteine is there, glycine, glutamic acid is there, glutamine is there, histidine is there, but isoleucine is there. Again, tyrosine, tryptophan, threonine. So, what happens now? You just look at this thing. Now, there is among the 20 amino acids, some is having a unique, that there is no C. So, C letter can be, C abbreviation can be used for this thing. So, histidine, there is no other H. So, H can be used, isoleucine I can be used, methionine M can be used, serine S can be used, valine V can be used. But still there is a problem with alanine, arginine, aspartic acid and asparagine. Now, among that, who will get the A? Who will get A? What is the basis? So, one of the problem was solved because of the unique first letter. Second part, then they studied that bioinformatics, which amino acid is most abundant or frequently present in the protein, and the first letter will go to that particular amino acid. So, that is the reason alanine here is with A. Okay? You see cysteine, glycine is there in G, but glutamic acid is not there. Leucine, lysine is there, L, leucine got. Proline, phenylalanine, proline is there, proline is present in all bone, cartilage, and threonine, T, So most frequent on abundance and purine 5, 6 number, and 5 number, 11 numbers are covered. Now how to take care of the rest of the things? Okay, so now you look at the emphasis on phonetic. How do you pronounce it? Arginine. Arginine. R is there. So that's why the R was there. So phenylalanine, how do you utter? Phenyl, F is coming. So that was the reason why phenylalanine, F was there. After that, there was a problem that still others could not be solved. And just I got a recently a small video I would like to show. Video double click just listen, enjoy the last part. I'll end here because see arginine, phenylalanine, this phentolamine, this part was covered. This one, that is phenylalanine was done, arginine was done. Aspartic acid, because of the pronunciation effect, aspartic acid. So D emphasis was there, that's why its name was D. Asparagine, the N emphasis was there, that's why N was taken. Glutamic acid, a E emphasis was there. Glutamine, just like a cutamine, just something like that pronunciation, that's why there is a cutamine. And then tryptophan, Y, W emphasis was there. Tyrosine, Y was there, that's why it was Y. A problem ended with the last one, K. How to solve it? Now, there are 26 alphabets, 19 have been covered somehow, and then K was left with. So, nearest letter, what has not been covered in this was taken, and out of this thing, if you say KLM is the nearest one, so L was covered with leucine, M was covered with M. So, only left out was K, and that is the reason K was taken for that. So, you understand what is the basis of single letter. Now, with this single letter, that 3 letter abbreviation can be compromised with 1. So, 3 amino acids can be coded. So, into the small space of computer card, punch card, that can be stored as an information. So, during that period of time, because of the paucity of the facility, I must salute that lady. She has done a wonderful job in the year 1966, which you cannot think of. So, these are the basis of single letter abbreviation, and as I told you, KLM, this is the nearest letter to the first letter of the amino acids among the 26 alphabets were taken. These are the 9 things, these are the 9 points, among which F, you remember that phenylalanine F508 mutation and KDL sequence, for which these 4 amino acids initial, at least you try to remember, this can come as an MCQ. Most of the questions from these amino acids, they will be asked during a viva, which I could not cover because of the paucity of the time. They will ask you what are the different types of amino acids, name one amino acids, or this type of, at least one question might be set so that you cannot answer to this question. What does the D represent? What does the F represent? So, this you have to memorize at least before the examination, before entering the hall, just remember this one, and thereafter you forget that much suggestion I can give it to you. So at least remember, try to remember whatever way you can is possible, at least these four. The rest of the things, at the end you can remember, I mean it's up to you. So you understand the basis of single letter for just to whatever way I told, that is the minimum amount possible from collected from different sources. I mean, you imagine in 1966 how much effort she took to do this job.